Good afternoon, everybody. I'm very happy to welcome you to the 21st D. Harold COPP lecture. Before we start, I'd like to first acknowledge that this lecture is held on the traditional, ancestral, and unceded territory of the Musqueam people. The COPP lecture is a Faculty of Medicine lectureship and supported by generous gifts from the Medical Research Council of Canada, the precursor of the current CHR, and also a generous gift from the Wolf and Gita Church Foundation. COPP lecturers are chosen by the COPP Lecture Committee, and I want to acknowledge who come from across the Faculty of Medicine, and I'd really like to give my thanks to them for their continued service and support of the lectureship. I also invite you, everybody is welcome to nominate COPP lecturers, and I welcome you to contact me or any member of the committee to nominate future COPP lecturers. I also take the occasion to thank Dean Dermot Kelleher and Head of CPS Ed Moore, who are coming to the end of their terms of service, who have been really very supportive over their past 10 years, I guess, of the COPP lecture. And I really hope, really appreciate that, and really thank you both for your support. This year, I'm very happy to welcome, Dr. Noboru Mizushima from the University of Tokyo, and as our 21st COPP lecturer. But before we get started, I'd like to invite Ed Moore, Head of CPS, to say a few words on the contributions of Dr. COPP to UBC and to Canadian science.
Thank you, Robert. Good afternoon, everybody, and welcome. And I'd like to tell you that Dr. COPP graduated from the University of Toronto Faculty of Medicine with an MD in 1939, winning the faculty's gold medal, which made him the highest ranked graduate of the Faculty of Medicine during that year. He then moved on to the University of California, Berkeley, where he completed his Ph.D. in biochemistry in 1943, and that began his long interest in bone and mineral metabolism that would be the focus of his research throughout the rest of his career. He then joined the UBC and the brand new Faculty of Medicine 75 years ago in 1950 as the inaugural head of the Department of Physiology, a position he held until his retirement in 1980. In 1961, he reported the discovery of a new hormone, calcitonin, so named because it regulated the level or tone of calcium in body fluids. In humans, it's the salmon form of calcitonin that's the most potent, and it's this preparation that's used for the treatment of osteoporosis, Paget's disease, hypercalcemia of malignancy, and is a powerful and non-addictive analgesic. Throughout his career, Dr. COPP received national and international recognition for his research and service to the medical research community. Most notably, Dr. COPP was a Companion of the Order of Canada, a select group limited to just 150 members at any given time. And in 1967, he received the prestigious Gairdner Foundation Award. And in Faculty of Medicine, he inspired generations of medical students with his lectures and laboratory demonstrations. He even continued with that well after his retirement with a lecture to the medical and dental students with a lecture on calcium homeostasis. In 1966, during a visit to UBC, the president of the Medical Research Council, Dr. Henry Friesen, announced a generous gift to the university to establish the annual D. Harold COPP lectureship. This was, as Robert has said, subsequently expanded with an equally generous gift from the Wolf and Gita Church Foundation. And in making his announcement, Dr. Friesen said, and I quote, "On behalf of the Council, I wish to recognize the truly extraordinary and varied contributions Dr. COPP has made, not only at UBC, but throughout Canada and indeed internationally. He has an outstanding record of accomplishment as a basic scientist, as a skilled administrator, as a teacher, mentor, and as an advisor to many. There would be very few individuals in the roster of heroes of Canadian medical scientists whose influence and achievements have been so wide-ranging. I wish to recognize particularly his role as a key member of the Farquharson Committee, whose report in 1959 led directly to the creation of the Medical Research Council, now the Canadian Institutes of Health Research. It would be my wish that the lecturers chosen would exemplify the sterling qualities of character and the commitment to scientific excellence and service that were so evident throughout Dr. COPP's distinguished career. Dr. COPP was indeed one of Canada's most honored scientists. He was a respected colleague and a remarkable ambassador for UBC, for British Columbia, and for Canada. Thank you.
Thank you, Ed. It is now truly my pleasure to introduce Noboru Mizushima from the University of Tokyo to UBC as our 21st COPP lecturer. Noboru received an MD Ph.D. from the Tokyo Medical and Dental University in 1996. He then went on to postdoc in the lab of Nobel Prize winner Prof. Yoshinori Osumi at the National Institute for Basic Biology in Osaka. In 1998, Noboru published a truly seminal first author Nature paper using yeast genetics to identify the protein conjugation system that underlies autophagy. He started his lab at the Tokyo Metropolitan Institute of Medical Science in 2004 and is now professor and vice dean of the Graduate School and Faculty of Medicine at the University of Tokyo. His research is focused on deciphering the physiological role of autophagy using mouse genetics and the molecular and mechanisms of autophagy in mammals. Most recently, his lab has done some really innovative and interesting work showing that sheet-like liquid condensates form on the surface of mitochondria and mediate mitophagy by Parkin ubiquitin ligase. He's also done some very nice work characterizing ubiquitination of lipids. And this is really beautiful work, and I've noticed that. But I just saw that what Noboru is going to talk to us about today is much more his work on mouse models and using mouse models to study critical roles for autophagy in embryogenesis, neurodegenerative diseases, organelle degradation in the lens, and also multilamellar body formation in lung. And I should say that my first interaction with Noboru came when we were both very young at an American Society for Cell Biology meeting, where we had just published a paper where we described some multilamellar bodies in cells and a role for autophagy. And he came to me and says, no, autophagy is not involved. Our autophagy knockout mouse has multilamellar bodies in the lung. And then I would say it must have been 20 years later, Noboru says, you were right. We knocked out the wrong ATG. And so I would only say for me, as someone who's not like an autophagy, aficionado, I think that only highlights the complexity of autophagy and its importance in fundamental cell biology. And so I would say Noboru and physiology. And Noboru is the 2016 recipient of the U. Hara Prize. He is the current president of the Japanese Society of Cell Biology. And he's also a Thomson Reuters Citation Laureate, which is awarded to researchers whose work is worthy of Nobel recognition based on the citation impact of their published work. And so I very much look forward to hearing Noboru's work on autophagy, and certainly I think we should give him a warm welcome. Thank you very much for coming, Noboru. Welcome. Thank you.
Okay, good afternoon. So thank you, Robert, for the very kind introduction. And I also thank the COPP lecture selecting committee members for selecting me as the speaker of this very prestigious lecture series. It's my great honor. So today I'd like to talk about autophagy and other degradation processes. And so first So first, I'd like to introduce what autophagy is. So autophagy is a kind of intracellular recycling system. So when autophagy is induced, a very small membrane sac elongates and encloses part of the cytoplasm, so resulting in the formation of this double membrane structure called autophagosome. So then lysosomes fuse with the outer membrane of the autophagal zone, and the lysosomal enzyme enter the very tiny space between the outer and inner autophagal zone membrane. So next, lysosomal enzyme degrades only the inner membrane and degrades the material inside autophagosomes. And finally, amino acids are delivered back to the cytoplasm and will be used for many different purposes. So autophagy is a kind of intracellular recycling system. So because this system is a bit complicated, I would like to show you a CG movie that was created by my previous undergraduate medical students. So now a part of the cytoplasm is engulfed by this autophagal zone. So sometimes intracellular organelles like mitochondria are also enclosed. So now this is almost closed. So then let's look at the inside of this autophagal zone. So as I mentioned, the autophagal zones has two membranes, the outer membrane and the inner membrane, and these membranes are connected at the rim. So now this rim is closed. So this very tiny neck is cleaved to separate into two membranes. So this is the closure of autophagal zone. So the closure step is mediated by membrane fission process, not membrane fusion. So once the autophagosome is closed, so lysosomes, typically more than one lysosomes fuse with a single autophagosome to degrade materials inside autophagosome. Okay, so this graph shows the number of autophagy papers published each year. So autophagy was discovered around 1960. So then in 1992, so this very young Dr. Osumi discovered autophagy system in yeast. And next year in 1993, so Dr. Osumi and his master students, Miki Tsukada, isolated 15 autophagy deficient mutants called APG mutants. And many people believe that this is the most important paper in the autophagy research field. And next year in 1994, a German group, Mike Thun and Dieter Wolf, isolated another set of autophagy mutants called AUT mutants. And in 1995, so Dan Klionsky in the United States also isolated a similar set of autophagy mutants called CBT mutants. And isolation of these autophagy mutants led to the identification of autophagy genes. Now they are called ATG genes. And so I was very lucky to be able to join Dr. Osumi's lab in 1997. So as you see here, so that was a very happy time for us because I didn't have to read many papers. So because there was almost no papers in the autophagy field. So what I should do was just doing experiments without reading papers. So, but now I'm very sorry that so you have to read many, many papers. So I think that more than 100 papers are published every week in this field. Okay, so to visualize autophagy, so we have used autophagosome marker protein LC3. So these are mouse fibroblasts expressing GFP LC3, and these small dots represent autophagosomes. So under basal conditions, so there are only few autophagosomes in these cells. But after amino acid and serum starvation, the number of autophagosomes increases to maybe up to 100 or 200 per cell within 30 minutes or 60 minutes. So autophagy is really very dynamic process that responds to starvation. So at a higher magnification, so you can see the formation of autophagosome like this. So it takes about 5 to 10 minutes. Okay, so we applied this method to study autophagy in mice, by generating GFP-LC3 transgenic mice. So these are in adult mice. So we found that autophagy is induced almost all tissues except the brain during starvation and after birth and also after fertilization. And we also observed that autophagosomes accumulated in many disease settings like acute pancreatitis. Okay, so this slide summarizes the function of autophagy. So it can be roughly classified into two groups. The first function of autophagy is to produce amino acids and other degradation products. So this function is important for the adaptation to starvation and also for early embryogenesis and before implantation. And the second function of autophagy is, of course, intracellular clearance or quality control. So autophagy can degrade many abnormal proteins and damaged organelles and even invading bacteria. So this second function is very important to prevent neurodegeneration and tumorigenesis. And so autophagy also has a very specific function in lamellar body maturation. So typical lamellar bodies can be found in epithelial cells of the lung and swim bladder in fish. So lamellar bodies contain lipid surfactants. So this is a protein lipid complex. So when surfactant is secreted, so it forms a very thin lipid monolayer on the water-air interface to reduce the surface tension. So then the lung and swim bladders can be inflated. So we found that autophagy is also important for maturation of lamellar bodies. But I should emphasize, this is not the first report. Actually, 20 years ago, so Dr. Robert Nabi reported that this is a very important paper. So he used the cell culture system and they found that autophagy is important for biogenesis of lamellar bodies. And 20 years later, so we happened to find the same thing in zebrafish. So when we make zebrafish autophagy knockout models, so we found that in a white-type zebrafish, this is a swim bladder, which is inflated. But in autophagy gene knockout, this is 1500 is one of the autophagy genes. So in autophagy deficient mutant, so lamellar body is there, but it is not inflated. So this is a white-type fish swimming in a cuvette of a spectrophotometer. So they can swim very well because they are fish. But, but when we look at zebrafish, 1500 knockout fish, so they cannot float up. So this is because the secretion of surfactant should be insufficient. So we also found, we also made type 2 alveolar cell specific 5200 knockout mice. This is Y-type neonates. So in contrast to Y-type, the autophagy knockout neonates show gasping respiration and became cyanotic. So this should also be due to insufficient production of surfactants. So, Robert, you are right. And so most of these studies have been done in mice, but how autophagy relates to human disease has also been elucidated. So in collaboration with Dr. Matsumoto, so we identified mutations in one of the autophagy genes called WIPP4 in a very rare neurodegeneration called SENDA or BPAN. So during early childhood, so the patients show only non-progressive psychomotor retardation, so they can walk. But after adulthood, the same patients suddenly developed very severe dystonia and Parkinsonism, and they became bedridden. And we confirmed that autophagic activity is partially reduced in patient blood cells. So we think that autophagy defects may contribute to pathogenesis of this disease. And also mutations in other autophagy genes called ATG, such as ATG5 or ATG7, also cause neurodevelopmental disorders in humans. And so today I'm not going into details, but the relationship between autophagy and human diseases is a very hot topic in this field. And many autophagy-related genes also associate with mostly neurological disorders. And this is just advertisement. So due to time limitations, I will not explain details. But if you want to measure autophagic activity in cultured cells, I highly recommend using this allotag-based autophagy flux reporters. This is a very quantitative and very objective method to study autophagy flux. So this is already published, and you can get reporters at Gene. So this is highly recommended. Okay, so today I'd like to talk about three recent topics. The first topic is autophagosome-lysosome fusion. Okay, so as I showed you, so lysosomes can fuse with autophagosomes, but this is only with matured closed autophagosomes. So lysosomes never fuse with unclosed intermediate structures. So if lysosomes fuse with these unclosed structures, lysosomal enzymes may leak out to the cytoplasm, which should be dangerous. So lysosomes should fuse with only matured, a closed autophagosome. The question is, why can lysosome recognize only matured autophagosomes? So this is the question. So more than 10 years ago, so we identified autophagosomal snare protein, syntaxin 17, so which interacts with lysosomal snare protein VAMP8 and SNAP29 to mediate autophagosomal lysosome fusion. So this is maybe okay, but very interesting thing is that syntaxin 17, so it has, although it has two tandem transmembrane domains, it can be soluble in the cytosol, and it localized to only mature closed autophagosomes. So this is very, very strange. So I want to show you the evidence. So this is LC3 as an autophagosome membrane marker, and this is a snare protein, syntaxin 17. So this is some closed mature autophagosome, which should be ready to fuse with lysosomes. So please look at this very small structure, which should be at very early stage. So now this membrane elongates, and now it is in the cup-shaped, but still syntaxin 17 was absent on this membrane. So let's please take carefully look at these structures. So now it is closed. So then syntaxin 17 is recruited to this membrane, okay? So when autophagosome is closed, this snare protein is recruited. So it can explain why lysosomes can fuse with only mature closed autophagosome, because the fusion machinery is only there. But the question remains, of course, how syntaxin 17 recognize only mature autophagosomes. Okay, so my previous PhD student, Saori Shinoda, found a very interesting phenomenon. So syntaxin 17 has a very short C-terminal region following the two tandem transmembrane domain. And if she deleted this C-terminal tail, syntaxin 17 can no longer associate with autophagosomes. So this C-terminal tail should be very important. So this C-terminal region contains many basic amino acids like arginine and lysine. And so when she replaced these basic amino acids, positively charged amino acids with alanines, the autophagosome association was impaired accordingly. So these positively charged amino acids should be important. Okay. So then we speculated that syntaxin 17 may favor negatively charged membrane. So we first tested this hypothesis using in vitro system. So we just mixed syntaxin 17 with liposomes with a different composition of phospholipids. And syntaxin 17 can associate with liposomes with containing negatively charged lipids such as phosphatidylserine and PI3P or PI4P, but not associate with the neutral liposomes containing PE or PC. So this data suggests syntaxin 17 indeed prefer negatively charged membrane. So what about in vivo in cells? So to monitor the surface charge, so we used membrane surface charge biosensors, which were already published. So these reporters contain different numbers of basic amino acids and phosphorylation site. And these reporters bind to membranes with different membrane charges. So first we use a reporter for weakly negative charges. So this reporter associates with the both unclothed autophagosome and closed autophagosome. So even before syntaxin 17 recruitment. So it suggests even unclothed autophagosomes may have weakly negatively charged. Then, so we use the reporter for strongly positive, negatively charged membrane. And this reporter binds to only closed autophagosome, but not unclosed autophagosomes. So these data suggest that closed autophagosomes are more negatively charged compared with unclosed autophagosomes. So then the next question is, what makes autophagosomes negatively charged? So we speculated this must be due to some change in lipid composition or lipid modification, because this is a very quick and robust change. So we screened a lipid species and found that PI4P, phosphatidylinositol 4-phosphate, is highly enriched only in mature closed autophagosomes, not unclosed autophagosomes. In this experiment, we used a CERT domain, a Ph.D. domain of CERT, but we obtained essentially the same result using other PI4P reporters. So PI4P is enriched in mature autophagosomes. Then finally, we tested whether PI4P is indeed important for syntaxin 17 recruitment. So to this end, so we used, again, in vitro system. So we purified autophagosomes from cells and mixed with recombinant syntaxin 17. And syntaxin 17 efficiently binds to purified autophagosomes. But when we treated autophagosomes with PI4P phosphatase, SAC1, the syntaxin 17 couldn't localize to autophagosomes. So it suggests that PI4P is indeed important for recruitment of syntaxin 17. Then we finally confirmed it by MD simulation. Okay. Maybe the okay. So we just tried MD simulation. So this is a membrane with PI4P, and the transmembrane domain of syntactin 17 can be inserted into membranes only in the presence of PI4P, not in the absence of PI4P. So again, this data suggests PI4P is indeed important for syntactin 17 recruitment or membrane insertion. So this is the summary. So we found that PI4P is enriched in mature autophagosomes, and so this, which should be important for the syntactin 17. And so we propose the electrostatic maturation model. So because autophagosomes become more negatively charged during maturation. And this model is consistent with a previous report published by Beth Levin and Ellen In's lab. So they showed PI4P is important for autophagosome lysosome fusion. But now we found that PI4P is an important regulator of membrane charge rather than a specific molecular signal. And this is also consistent with a recent report published by Gaba Yuha's group. So they also show PI4P is important for syntactin 17 recruitment in just published last year. Okay, so in the second part, so I'd like to talk about our unpublished stories about the role of autophagy in the brain. Okay. So this part was done by our very talented assistant professor, Tomoya Eguchi. Okay, so again, almost 20 years ago, so Masaaki Komatsu's group and our group reported made brain-specific ATG knockout mice, and these mice developed neurodegeneration or neuronal dysfunctions with protein aggregates and abnormal organelles. So this is already known. So what is unknown is so whether these neurological dysfunctions can be reversed when we restore autophagic activity. So we generated reversible ATG knockdown mouse model. So we decided to use the FAST system, which was developed by Dr. Kenji Tanaka at Keio University. So this is a very useful system. So we inserted the FAST cassette containing TET-O element, a tetracycline operator element, into the first non-coding exon of ATG101. So ATG101 is another essential autophagy gene. And we closed them with another transgenic line expressing TTS, tetracycline-dependent transcriptional silencer, under the control of ubiquitous promoter. So in the resulting mice, so TTS binds to the TET-O element and replaces the endogenous promoter of ATG101 to suppress its expression. So the ATG101 expression was suppressed and autophagy is also suppressed. But when we treat with these mice with doxycycline inhibits the function of TTS, now allowing the endogenous promoter to drive ATG101. So autophagy is turned on. So we can turn on the autophagic activity on and off in tetracycline-dependent manner in mice. So we check the autophagic activity in the brain of these mice. So in the presence of doxycycline, so ATG101 was normally expressed, and P62 is a typical autophagy substrate, and P62 didn't accumulate. So then we removed tetracycline, and after 4 days, the levels of ATG101 was decreased, and P62 started to accumulate. And at 14 days after doxycycline removal, ATG101 was completely shut off, and the P62 highly accumulated. So autophagy was efficiently knocked down. So then we treated these mice again with doxycycline, and surprisingly, only after 4 days, the levels of ATG101 and P62 returned to almost normal levels. So suggesting that this system works very well in the brain, and also suggests that substrate degradation by autophagy is very efficient in the brain. Okay, so we analyzed the phenotype of these mice. So first we checked their survival. So after 2 weeks of doxycycline removal, these mice started to die. And after 4 to 6 weeks, most mice died. So we analyzed the reversibility of neurological functions during the first 4 weeks. Okay, so first we check the motor function of these mice using 2 famous tests, rotor-rod test and beam test. So first I want to show the result of the rotor-rod test. So we prepared 3 groups. In group A, autophagy was always on. And in group B, autophagy was shut off for 4 weeks and then rescued, turned on for 4 weeks. And in group C, autophagy was shut off for 8 weeks. Okay, so here is the result. So in group A, rotor-rod score was always good. No motor dysfunction appeared. But in group B and group C, so the rotor-rod score dropped during after doxycycline removal, so suggesting the motor dysfunction. But it was improved only in group B, so when autophagy is reactivated, but not in group C, so where autophagy remained shut off. So these data suggest motor dysfunction caused by autophagy defects can be reversed. Okay, so I will show the actual data movie. So this is before autophagy off, and this is after autophagy off, and this is after autophagy rescue. And the important point is that these are the same mouse at different time point. So you don't have to worry about individual variations. So you can compare these mice directly. Okay, so please look at this mouse after autophagy off. So they are trying to stay on this rotating bar, but now this mouse decided to rotate together with the bar. But the same mouse was able to stay on this rotating bar for much longer time. Okay, so the motor function was reversed. So we performed another test called the beam test. So here is a very narrow bridge. And this is before autophagy suppression. The mice can stay across the beam very smoothly. This is normal. And after autophagy suppression for 4 weeks, the motor dysfunction appeared in the same mouse. This is the same mouse. And after autophagy rescue, the phenotype was much improved. May not be 100%, but it was much improved. So again, these are the same mouse at different time points. Okay, so next we check, this is a quantification result. So the time to cross the beam was extended in both group B and group C, but it was improved only in group B. So when autophagy is reactivated. Okay, so we also check their learning or memory functions. Excuse me. Okay, so first, mouse was placed in a bright compartment, but so mice prefer dark places. And so here is the gate. So mice enter the dark places when the gate opened very quickly because mice like dark places. So the entry time was very, very short. But unfortunately, so mice received electric shock in this dark room. So they realized that this is a very dangerous place. So we repeat the same experiment after 48 hours. So now, so mice wouldn't go into the dark places even after a gate opened. So because they remember that dark place is very dangerous. So the time to enter the dark compartment was extended. So basically, they didn't enter the dark places within 5 minutes. So this is before autophagy suppression, normal mice. And we did the same experiment after autophagy suppression, and the time to enter the dark places was still very short, even after they received electric shock in the dark places. So they didn't remember the dark places dangerous. And so when we used the same experiment using rescued mice, the entry time was again prolonged. So this indicates the learning or memory function can be also be reversed by autophagy restoration. And we also performed the radial arm water maze test. So in this test, so we trained mice for 2 days, and in the test, so we counted how many arms mice try to find the goal. So here is the goal. So after 2-day training. Okay, so this is before autophagy suppression. So mice quickly reach the goal. So they try just 1 or 2 arms to find the goal. But after autophagy suppression, so they tried many, many arms. And so it's very confused, and finally reached the goal. So the cognitive function was impaired. So they didn't remember where is the goal. But we used the rescued mice. Again, they quickly reached the goal. So the cognitive function can be reversed. Okay, so we also checked the histology. So we didn't observe significant cell death after autophagy suppression, 4 weeks of autophagy suppression. So these are Purkinje cells in cerebellum, so which should be most sensitive to autophagy suppression, but we didn't see significant cell death. And but by electron microscopy, we found these are axons of Purkinje cells. So these axons are significantly swollen. And we observed many membrane structures in these swollen axons like this. And after autophagy rescue, the phenotype was again much improved. So still we observed some swollen axons, but the quantification reveals that there is a significant improvement. And also we checked proteostasis. So before autophagy suppression, we didn't observe any protein aggregates or inclusion bodies in the brain. But after autophagy, 4 weeks of autophagy suppression, we observed many protein aggregates everywhere in the brain, so such as cortex, hippocampus, and cerebellum. But these inclusions were all cleared after autophagy restoration. So again, autophagy is very efficient to clear abnormal proteins. So we also check the whole proteome. So when we compare the proteome before autophagy suppression and after autophagy suppression, so we found many proteins accumulated after autophagy suppression. So these are autophagy substrate and autophagy adapters. So besides these autophagy-related proteins, we also found so many proteins in the organelles, such as the ER and Golgi, also accumulated. So this is consistent with the electron microscopy observation that many membrane structures accumulated in swollen axons. And when we compare the proteome after autophagy suppression and after rescue, so these proteins are mostly degraded after autophagy rescue. And accordingly, when we compare the proteome before suppression and after rescue, so there is almost no change. So suggesting that autophagy normalized the proteome almost completely. So again, autophagic degradation was very powerful. And finally, we wanted to check whether the phenotype we observed was indeed neurogenic, because tetracycline can also regulate other tissues, such as muscles. So to this end, so we removed the tetra element only from the brain using the nesting cream mice. So we called these brain rescued mice. So in these mice, TTS can no longer function only in the brain, but still functional in other tissues. So as I showed you, the systemic knockdown mice died by 6 weeks of doxycycline removal, but brain rescued mice can survive for much longer time. So early death may not be rescued. So this may be due to damage of the intestine or infection, but the survival rate overall survival was much improved. And these brain rescued mice can stay on this rotating bar even after 8 weeks of doxycycline removal. So these data confirmed that part of premature death and motor dysfunction are indeed neurogenic. Okay, so this is the summary of this part. So we established reversible ATG-101 autophagy knockdown mice, and neurological dysfunction and abnormal morphology caused by autophagy defects can be reversed by autophagy restoration. And restoration of autophagy cleared the inclusion bodies and normalized the proteome. So based on these findings, so we speculate that as long as neurons are alive, autophagy activation may restore neuronal function, even after phenotype appearance. Okay. So in the last part, I would like to something beyond autophagy. So generally, it is believed that intracellular organelles are degraded by autophagy. So this is a mitochondrion, and this here is a fragment of ER. So these are degraded by autophagy. So, but I would like to show you a completely different type of organelle degradation. So that occurs in the lens. So the major contributors are Hideaki Morishita, former assistant professor, and again, Tomoya Eguchi. So the lens of the eye is a very transparent, beautiful tissue. This is transparent. So you can see this slide because your lens is transparent, I think. But lens cells, fiber cells, originally contain a lot of intracellular organelles, such as mitochondria, EL, and nuclei. But these organelles are degraded from the center to the periphery during differentiation, so making the lens transparent. So although this is a very dramatic process, its mechanisms have been largely unknown for more than 100 years. So these are mouse lenses. This is a lens from embryonic day 17.5. So you can see the green signals the EL, and red signals indicates nuclei. So there are many organelles. But only after 2 days, so these organelles were degraded in the central region. forming the organelle-free zone. So cells are there, but they don't have organelles. So initially we thought, so these organelles must be degraded by autophagy. So because we like autophagy, but we were wrong again. So when we look at the lens from autophagy knockout mice, these organelles were normally degraded. So with no problem. So this data suggests this is independent of autophagy, and there should be another great system to degrade these organelles in the lens. So we have been working on this project for more than 20 years, and finally we reached the conclusion. So we use zebrafish as a model, and this is a transgenic zebrafish expressing GFP in the EL matrix and RFP in the mitochondrial matrix. So these are lens, and we can directly observe lens differentiation on the stage of the microscope. So during differentiation, so these organelles were degraded from the center to the periphery, forming the organelle-free zone. But we realized that these signals became diffused, not just degraded. So it means these organelles are ruptured, not degraded inside lysosomes or autophagosomes. So what is the mechanism of this membrane rupture? So we performed CRISPR mini screen. So we collected about 60 genes that are highly expressed in the lens and performed CRISPR knockout screening. And we injected guide RNAs into one-cell embryos and directly observed their lens just 3 days later. Then we identified one lipase, PLA81. This is a phospholipase A acyl transferase. So this is a cytosolic lipase, not lysosomal lipase. So this is a white-type fish. So in white-type fish, mitochondria were degraded between 50 to 70 hours post-fertilization, and EL was also degraded. But in plat knockout fish, mitochondria degradation was suppressed, and EL degradation was also suppressed. So this single gene plat is essential for organelle degradation in the lens. So this gene is conserved in vertebrates, and mice have 3 homologs, plat 1, 3, and 5. And among them, only plat 3 is highly expressed in the lens. So we generated plat 3 knockout mice, and this is a control experiment. So in control mice, these organelles are degraded in the center of the lens. But in plat 3 knockout mice, these organelle degradations was completely suppressed. So this system is conserved in mammals. And the lenses from plat 3 knockout mice were less transparent, suggesting that this plat-dependent organelle degradation is important for optimal lens transparency. Okay, so what is the mechanism? So plat has, of course, a lipase domain, and it has a C-terminal transmembrane domain. But again, this protein is very interesting. So it can be soluble in the cytosol. But during lens differentiation, it forms punctate structures in these lens fiber cells. So this is a magnified image. So this is a mitochondrion in the central region. So initially, in the beginning, plat was not there, but during differentiation, plat enzyme was translocated to this mitochondrion and degrade them. So again, plat was recruited, mitochondria were degraded. So these are frames of this movie. So when plat enzyme lipase was recruited, mitochondria were degraded within 1 hour. So again, this is a very efficient system. So I don't show the data, but lipase domain is of course important, and this transmembrane domain is important for organelle targeting. So this is already published. So I want to show just summary of this system. So we found that cytosolic lipase plat is required for organelle degradation in the lens. To recruit the plat enzymes, we found that these organelles have already partially damaged. So these organelles have small membrane pores, and these pores can recruit plat enzymes. Specifically, the transmembrane domain recognizes these small pores. So currently, we don't know how these small membrane pores are generated, but we found the upstream transcription factor called HSF4 is required for both membrane damage and plat recruitment. So because HS4 is activated from the center to the periphery, it can explain why membrane degradation occurs in this order. But currently, we don't know what happens downstream of HSF4. But so this system is very different from canonical autophagy. So in canonical autophagy, organelles must be enwrapped by autophagosome one by one. So this may be not very efficient. So to degrade everything, so this new system should be more efficient. So, but now we are also studying so the insertion mechanism of plat to membranes, but this is still ongoing project. And recently we found a very interesting paper. So the fish, these are deep sea fish or cave fish. So they lost plat1 enzymes genes, or this gene was heavily damaged in this fish because the eye function is not important for this fish. So this data also support the idea that plat is very important for the eye function. Okay, so I'd like to thank everyone who contribute these studies. So again, Saori Shinoda contributes to the SNEA-dependent autophagosome lysine fusion, and Hideaki did the zebrafish analysis, and Tomoya is doing the reversible autophagy knockout mice, and Yuji contribute to MD simulation in my lab. Okay, so again, so thank you very much for the attention.
So I would. I would just like to thank Noboru for a really interesting talk on autophagy, but also on non-autophagy mechanisms of organelle degradation. And I would like to invite Dermot, Ed, and also Leonard to join me and to present a small gift, you'll see, gift to Noboru in honor of his presentation. And I'd also invite perhaps Dermot to say a few words. So a small gift. It's larger than normal. But this is perhaps, Ed, I will let you pass this on to Noboru. And I can just, maybe this is just, I'll say, before you say something, Dermot, I'll just say this is a carving that depicts a jumping salmon and was carved by Glenn Edwards, a member of the Coast Salish Nation, who was born in Nanaimo, Vancouver in '58. And we have a small plaque there that's going to be on the plaque that's in the hall of the LSI. And so please, Dermot, go ahead.
Thanks very much. Gosh, what a pleasure it is to hear you speak today and to hear you speak on the traditional ancestral and unceded territory of the Musqueam people. I'm saying that for a particular reason. because when I was a medical student, I think we became aware that there was a drug called calcitone, and that drug was useful in Paget's disease. And I won't tell you how long ago that was. Well, I was a medical student between 1972 and 1978, and that tells you something about the incredible influence of Dr. COPP in the first instance, because he and what an extraordinary synchronicity it is that he found that salmon calcitonin in these lands was the most potent form of calcitonin for use in humans. And it just goes to illustrate, I think, for anybody in the audience, you have to think very broadly when you're working in science, and think about what's happening in salmon as well as in humans, because many systematic approaches to human genomics have had their origins in salmon. Just to say, though, in respect to Dr. COPP, I think, again, just if we look back at the achievement of not only identifying a new hormone, but finding a new hormone that has a clinical application in osteoporosis, in Paget's disease, in bone pain and malignancy, an extraordinary feat of what we now call translational science, although it wasn't called that at the time. And it's just really important to think about history and tradition in the places where we live and work, because that's so evident in this work, and so important for all of us working at UBC to think back to the incredible achievements of Dr. COPP. I will just say a quick word about Noboru's amazing presentation, and I have to say my head is spinning from it, because the incredible demonstration of an on-off effect of an autophagy gene in terms of neurological deficits was absolutely amazing to witness, and begins to ask all kinds of questions, and begins to provide us with tools to answer all kinds of questions regarding the opportunity, the influence of autophagy in disease. But I thought the story about the lens is just incredible, isn't it? To think that we have a mechanism. I mean, how many people in the audience have thought about why a lens is clear? Anybody has thought about that incredible question? Why is a lens clear? And to hear this explanation of why a lens is clear, and begins to for me anyway, to provoke all kinds of interesting questions about evolution. And you showed the fish in the last slide, the deep ocean dwelling fish, but an incredible set of wonderful experiments which have illustrated the solution to a question that many people have never really asked, and it's just a testament to you, Noboru, and to the incredible importance of curiosity in science, and the incredible importance of thinking about the tools to answer a question in the most effective way. So thank you very much for an amazing lecture. I'm sure that all of the young people in the audience have got so many new ideas as a consequence, and we really welcome you to UBC.
Thank you, Dermot. Thank you, Noboru. I never thought of the salmon as a model organism, but perhaps we should reconsider. I would like to open the talk for questions. And if anybody is on Zoom who has a question, please feel free.
Can I ask you a question?
Yes, absolutely.
Yeah. So I just wanted to ask you a question about the lens work, and where do those genes come in on an evolutionary sequence? Because you refer to the loss of the gene in the deep living fish, but where do they appear as we go through evolution?
Yeah, the system is present only in vertebrates.
Only in vertebrates.
Yes.
Yeah. Okay. Thank you.
Questions?
Thank you for the wonderful talk. I have a question about your brain knockout of autophagy. It looked in the electron microscopy images that you showed that not only were the axons much larger, but that there was also a loss of myelination. Have you quantified that? Is that indeed the case? And is there cell death of other cell types besides neurons?
Okay. Okay. Yes, that is very important question. And we observed that myelination is less in after an autophagy knockout. That is because the axon is swollen and just is diluting. So, but we haven't carefully quantified the thickness of this myelin. But yeah, we want to do that. And also, we also now checking the cell death in other cell types, but we haven't got very conclusive data so far. Okay, thank you.
Hilla?
Yeah, thanks for the great talk. I was wondering about PLAT1. You show that it has a transmembrane domain, but it seems to be very promiscuous. It goes to every membrane. I'm assuming that it has to go to ER, mitochondria, outer and inner. How does that work? One question. The second question is, what do you think about the harmful consequences that might be if you just rupture the membranes in terms of lysosomes, hydrolases, for example, and mitochondrial DNA?
Okay. So the first question is, so the enzyme, like the lipase, can go to any organelles once they have membrane damage, pores. But for example, if we simply express the lipase in HeLa cells, so nothing happens because all membranes are intact. But if the membrane is damaged, so it can go to any organelles, like mitochondria. For example, mitochondria during apoptosis, mitochondria has large pores, so blood can go there. So basically, any membranes with damage can be the target. The second question is, what is the consequence of membrane damage? Yes, that is another very important question. So we think that blood-dependent membrane rupture is just trigger of a large sequence. And I think that, for example, proteins can be degraded by the ubiquitin proteasome system after membrane rupture. And we don't know the fate of the ruptured membranes, but it may be solubilized by lipase, and also the lipase cleavage produces lysophospholipase. So lysophospholipase is also a kind of detergent. It can solubilize other membranes. So I think all these contribute to the final transparency of the lens.
Yeah, I have maybe a final question is when you showed the proteomic analysis, there were lots of membrane organelles there, but there were no mitochondria. And I'm wondering,
that's good. Yeah,
why
very
not?
good point. Yeah, very good point. So only ER and Golgi proteins accumulated, but not mitochondria. So I don't know why, but mitophagy may not be so active in normal neurons, but I don't know.
Okay. Great. Well, I think we should give Noboru a really warm, great thanks and warm welcome to UBC. Thank you very much for a really wonderful talk, really interesting science. And I'd like to invite everybody after the seminar to join us in the West Atrium for a social. And thank you again, Noboru.
Thank you.